Okay, so I welcome all of you to this edition of the NSF Wednesday Colloquium. And it's a pleasure to have Professor Ruchi Anand, who is joining us maybe just an hour away from this place, from her own office at IIT Bombay for this colloquium. And of course, before I introduce Professor Anand formally, who happens to be a dear colleague and a friend, I would like to sort of state for Professor Anand and the students as well as the audience members who are listening to us for the first time. The NSF Wednesday Colloquium is actually as old as the TIFR itself. It was started by Professor Homi Bhabha, who was our founding director. And the idea that he proposed to all the members of the institute right then was it would be a great idea if all the members of the institute, used to be a small institute at that time with physicists and maybe a few biologists and chemical physicists, to get together once every week. And the time and the date was, the day was chosen as Wednesday 4 p.m., where everybody get together in a lecture theater and listen to the experts from around the world who would physically come down to give a talk, to give a talk which has which can connect to larger scientific community and give a colloquium. So that tradition has been kept alive for all these years. It's almost like 75 years of this institute now. And it's actually a big honor for me to continue this tradition. And without any question, today we have a leading expert in the community of structural biochemistry, structural biology, Professor Ruchi Anand, who is going to talk about allosteric regulation of molecular tunnels. So before she goes into the colloquium, I'd like to just give a brief introduction. Ruchi actually did her bachelor's from Miranda House, Delhi University in 1996. And then this was in chemistry. And then master's again in chemistry from IIT Kanpur. And she finished that in 1998, after which she went to Cornell University for her PhD. After finishing her PhD at Cornell in 2004, she moved to, she did have 2 postdoctoral stints in the US, one in Sloan Kettering as a Howard Hughes Fellow, and then at University of Pennsylvania, where she spent almost a year and a half before moving to IIT Bombay and opened her own research lab, completely with such a mindset that she will do structural biology, crystal structure, crystallography of large molecular complexes in India and at IIT Bombay. And over the last roughly maybe 14 years, Ruchi, right? 13 years, she has successfully sort of established a world recognized program with all the efforts to bring in, the infrastructure that is needed to do structural biology in India. So, and of course, her research interest has spanned different kinds of, research topics, but most principally, she has been sort of interested in structural basis of antibiotic regulation and mechanism of resistance in bacteria. And as well as design of different sorts of biosensors by looking at the structural aspects of proteins and protein ligand interactions. And also in very recent times, she has been on search for molecular tunnels where, you'll have allosteric regulation, a work that recently she published in Science Advances, and she's going to talk about today. Ruchi, so without further ado,
thank you so much.
Nice if you could start the colloquium. Thank you.
Thank you so much, Yotishman, and all of you at TIFR. It's my pleasure to be here. I wanted to say that with an emphasis because I always feel very warm to be in TIFR, and I feel like I'm associated with TIFR in several ways, always. GK is here, who has been my constant collaborator. Even today, we are collaborating. and papers with GK are still coming through with us. So, and I'm sure we're going to have a lot of future collaborations with various other chemical and bioscience members.
And I just wanted to add to everything what I wanted to say, that Ruchi is one of our favorite colleagues across the country, and always an inspiration for many of the young scientists, because the way she disseminates, she talks about her science is something that we all look up to. Thank you, Ruchi.
Now Jyotishman is buttering me. I don't know why. Okay, Jyotishman, I will collaborate with you. Done. I'm just joking. Okay, let me start my talk. Let's go to the science behind it. So today I'm going to be only talking about one system. I decided that I have given departmental talks in the past, and there have been multiple topics, and I feel I will only concentrate on one topic today. And but I do want to tell in my opening slide that we work a lot on antibiotic resistance, and this is something that we hope we have solved the structure of this ribosome with a methyl transferase, and these methyl transferases actually result in antibiotics to stop working because of the methylation. And we have tried to understand this process of how these methyl transferases find the antibiotic. This is one of the projects that we are pursuing, and then in our quest, which we are trying to submit this paper now, we realized that long distance allostery is at play even when this methyl transferase wants to find its own target within the ribosome, and bases which are like more than 20 angstroms away, they need to go in a specific pocket by flipping, so that at this red site can get methylated. So allostery is something which is around us in systems where you will see in biology, it's a very, very, recurring phenomena, I would say. And surprisingly, we are working with biosensors, and we try to develop biosensors for phenol, benzene, toluene. And in this system also, we are now pursuing the allostery. Initial work, we did a lot with just phenol binding. And trying to make a good biosensors. And now we are realizing that this binding of the sensor is coupled to a readout domain, which is called, which is an ATP hydrolysis activity. And that's how we were reading out our sensors. Okay, so now we are using this in the side of application towards developing electrochemical sensing. That's completely different ballgame. And towards our allostery side, we realized that this ATP hydrolysis, the whole process, where phenol, the signal goes to the readout domain, is in the presence and absence of this biosensor. There is a huge conformational change and also oligomerization, which is like the very famous proteasome AA plus and proteasome and AA plus machines, which are there, very similar to that. The whole process is happening over here also. So here again, we are collaborating with Jagannath Mandal for simulation, Anand for trying to understand ATP association and allostery, to understand this biosensor system from an allosteric perspective. For our third, this is the project that I'm going to talk about today. And this project is not a recent thing. To be very frank, I got my PhD on this project. This is the protein that I solved the structure in 2004 at Cornell. My advisor had actually said that he would not give me a PhD. And later I found he was joking to me that if I don't solve the structure of this protein, and I did that at that point, not realizing that it was just a false way of getting me to work on it. But then what happened is that I left the lab, and these questions kind of remained. And I kept on telling him, why is nobody working on it? He says, nobody wants to work on your complicated system. I've been trying to get people. So many years later, when I came back to India, I decided to pursue this project again and, go ahead with it. So it's something that I wanted to solve the problem at hand because it was with me for such a long time. So that is how this project has been initiated. And it turns out that it is a project which involves molecular tunnels. So just to give you introduction about tunnels, there are many enzymes which have tunnels, and there are for different reasons in biology, tunnels are there. One of the reasons that tunnels are there are because you may have certain substrates which are so unstable that if you put them in the environment, in that cellular environment, they may degrade. Then the second reaction will not happen. It can also be that these are substrates which are so difficult to produce in such low quantities. You don't want them to be diffused out into the cell. It could also be that they are toxic to the cell, and you don't want the cell to ever see them. And let them be consumed at the site of production. So there are many reasons that these tunneling enzymes are there. For example, here, if I show you the carbamoyl phosphate synthetase is a very classic enzyme where it has a tunnel for ammonia and indole. It's 95 angstroms, and it's a pre-formed tunnel. The enzyme has this tunnel with various domains. So 95 angstroms, the molecules move from the start to the end to do their reactions. In the middle, they stop here and then continue. So you can see that tunnels are.
Ruchi, can I ask a question here?
Yes,
Just a very general question.
Definitely.
How does one look for these tunnels? In the sense, how do you know it's a tunnel? It's a molecular tunnel and not something for other purposes. That only one set of molecule can go in and not other molecules, or is it?
Okay, I'll try to answer this question. There can also be tunnels which go to the substrate binding site. For example, in many enzymes where you have oxygen as a substrate, you can have tunnels which are there, which go to the active site from outside.
Right.
Okay, and so there can be outside connecting to the center of the group or wherever the substrate is, the active site is, and there can be internal tunnels. When I'm talking about these molecular tunnels, they're within the enzyme without ever having to come out at all, not from surface. Okay. Now, how do you find them? Is that there is a software called Kword. Okay, this is a simple way of finding them where you look for spaces and you can give the radii that you want, and then you will be able to tell what kind of molecule can pass through. So you can set your radii, and according to that, you can search whether this molecule can go through this space or not.
So you basically have a sphere that can pass through, you can traverse the sphere.
And they can we keep on doing this. And there are many other methods. Kword is just one I'm telling you because we use this software called Pymol, and you can plug in with it. But there are many other softwares people have come up with where they can actually, then basically what they want to do is they want to look for spaces with a certain radii.
And the other question I had is it irreversible if the substrates go inside? It will have to go in to the end of the tunnel, or it can also come back out? Is there a gradient? Is there
in our system, there is leakage. Okay? So that means it is leaking away from the tunnel and it is not like going. But I will tell you something exciting that we have found out, which we need a lot of experimentation to confirm though. Okay, and that I didn't want to give it away, but there is something which may sometimes be pushing the, for example, okay, let me say in ion channels, for example, they have these hydrogen bonds, which kind of, pull the substrate. In our case, it's substrate can be as a proton, sodium, potassium, or it can be proton pumps also. They can be pulled if they have a charge. But the tunnel that I'm going to talk about doesn't have any charge molecules, so they are more harder to control because you don't want to get them to be chemically changed. Yet you want them to reach the other side. So how do you do that? Okay, so I will talk about it a little bit what we think and some future experiments which we are trying to do little bit. And any thoughts on it would be very good. So now when I talked about tunnels, what if you go across many tunnels at systems which have tunneling, and I'm here little bit focused on ammonia tunneling because I'm working on it. But there is, like I said, there are lots of other type of tunnels which are there, and they don't have to be internal tunnels also. So many of the tunnels, when people find them, they find that the tunnels generally come with a lot of controls because something has to be produced at one site and it is many times going to the other side, the second. When it is produced, should I keep the tunnel open because water may enter or something else may enter? You don't want. So there are like many checks and controls and balances in the tunnels, and they open and close, and they are called gates. Okay, there are many kinds of gates. If you read reviews on these tunnels, there are gates which are like aperture gates, where they open like this, together, many residues. There are gates where they flip it out like this. Okay, single gates, single amino acids. There can be gates which open like this. Just imagine, all those things that you see in your life, the gates that are there. So there are many, many kinds of gates, complicated gates.
Ruchi,
Yes.
can I ask a question?
Sure.
So this opening and closing and regulation, what regulates the opening, closing, or the aperture, the different things that you're saying, flipping? How does this thing get regulated?
So today I will talk about that in my talk, in my system. But each system has its own unique way of telling that how to open and close. So there's a lot of allostery because of that, because many times what happens is that there is this talk in one of the, if suppose there are two active sites, they have to know the substrates have entered. Somehow the information has to pass. And generally it goes via an allosteric network. So many of these enzymes, for example, you can see this tryptophan here, it moves out of the tunnel. Okay. And the signal is coming from somewhere else. So they have the full connections are there in these enzymes. And you can go for each enzyme and it will be a detailed connection network that you can develop actually. And so that's what although I should not go back, we were trying to do these allosteric networks that connect amino acids. And many times they are not obvious to us when we first look at them. And there are also papers which tell you that sometimes allosteric networks are not even conserved. Alternate allosteric paths can also develop. So it is very confusing many times to see, but in many cases, very, very strong allosteric cues are generally conserved. There can be in the middle some which may be variable.
So you're saying it's an internal protein-protein interaction that is basically governing whether
protein hydrophobic surfaces, hydrogen bonding networks. I will. My whole talk is about this only, how this enzyme does it. So you will get to know a little bit more about it. And there are other systems which analogously do in different ways. They have their own way. In some cases, a domain moves. In some case, nothing moves, only hydrogen bonding networks change. That can be static allostery, So there are many ways in which they try to communicate the long distance, what is happening. So one of the ways is physical movement of gates where the channel is actually blocked, and when whatever the information they get in whatever manner I will describe for our system, they move out. Okay, and they let the substrate pass. Otherwise, they don't want, in the absence of the reaction being in its correct sequence, for anything to come in and go out. Okay, so there can be at the top, at the bottom, and even in the middle, depending on the system. Some systems have less checks and controls, some systems have more checks and controls. Okay, so it also depends on how important that reaction is, and do they use it for some other purpose or not. So the enzyme that we are going to be talking about today is this enzyme, which is a core enzyme in the purine biosynthetic pathway. And in the purine biosynthetic pathway, this particular purine ring formation is the job of this pathway. So first, the 5-membered ring is formed from pyrophosphate to this 5-membered ring, and then it keeps on building. And there are actually 3 tunneling enzymes here. 2 of them are in the first step, first and the 4th step, and there's one in the latter step. So I'm going to be talking about the 4th enzyme. This is just before the closure of this 5-membered ring. So let me show you the reaction. So ultimately, this is what is going to be formed, which is precursor of DNA and RNA, very important pathway. This pathway you disrupt, death for us, for bacteria, for everyone. Okay. So you can't play around with this pathway. Actually, in humans, this pathway, many enzymes are fused to make sure that this pathway is functioning. It's a very, very complicated biosynthetic pathway to look in various organisms also, very interesting. So the reaction that we are looking at is, like I said, the formation of this 5-membered ring, not happening just before that. This C double bond O is called FGAR, formyl glycamide ribonucleotide, will become just the incorporation of nitrogen happens here, C double bond NH2 plus, which is formyl glycamidine ribonucleotide. And this happens with first reaction. There are 2 reaction sites. Reaction 1, there is an active site where glutamine gets converted to glutamate and ammonia. Then in the reaction site 2, C double bond O becomes a good leaving group with the help of ATP. You can see here. And then ammonia tunnels 25 angstroms inside. Okay, and reaches this binding site. Ammonium is not a good substrate because it's not a good nucleophile, so it has to be in its ammonia form. So this is the prerequisite for this particular reaction. Okay, so what we did was that several years ago I solved this structure, and when I saw this structure, I was amazed because it's 140 kilodaltons. Most two reactions enzymes can be 60 or 70. This is huge because you can see this blue area, it is gene duplicated. In this green area that you see is where the you will actually see this formation of this glutamine binding site. And here is where I'm pointing is where the active site is. There is another site which is ADP binding site, which I still don't know actually what it does till date. I'm still working. I mean, if you work on this enzyme, I think it's spent. And there is this adapter yellow domain, which nobody knows knew what it does. Now we have a lot of idea, but this ADP site is some sort of a regulatory site that we think, which is kind of the ATP, the product of ATP hydrolysis ADP. And so you can think that there is some kind of self-back regulation, and I still don't know the answer. So, but to give you in short, we had solved this enzyme and we got this huge structure with the enzyme in an inactive state, in the sense that the glutamine the cysteine is the attacking nucleophile, and cysteine had already bound it and made it thioester intermediate, and it's a catalytic triad with cysteine, histidine, and glutamic acid. Sorry. So this particular thing was trapped in the enzyme. It's like the enzyme, but there was no tunnel. How does this reach this site? We had no clue. Okay, and now I'm skipping to the details of all the years. When I first solved it, we didn't even know where the active sites were. We did not know whether this was the active site or this was the active site. We first thought this is the active site because ADP is bound, which is a product. And then we realized there is no space for anything else to go there. And over time, we figured out that this is the binding site. This is the active site second. And God knows what this is till date. But we have some idea, I'll tell you. But this site was people, me and then students after me, we tried to find species with this yellow, green, and this bluish domains are separate, and we could try to get some leads on it, and because that's how we know more about this active site. So
Ruchi,
yeah.
Ruchi, can you just go back to the slide of the chemistry where the ammonia comes in, just for a second? Yes, here we're here. So could you just tell me what is going on in the lower part? So you have the
so this C double bond O is here in the glycamide molecule.
Okay.
This gets converted to C double bond NH. Okay, and this is a very bad leaving group. I mean,
yes.
So I think it just gets phosphorylated by ATP. And then there is an intermediate is formed, and then the ammonia
the PI group leaves, the phosphate group leaves.
The ADP leaves and phosphate, and then the phosphate leaves, and then you get this ammonia here, and you get phosphate to leave.
Correct. Okay, thank you.
And then the ring closure will happen after that.
The next step.
Yes.
Okay, thank you.
I have a question, Ruchi. Hi. Can you hear me?
Yeah, I can. Yes.
Okay, so the last slide, you had the title. Why is this one so big? Yeah, why is it so big? I frequently look at any protein. I frequently ask myself the question. And so, what was the answer? I was looking for your answer.
Yes. I actually I would be happy to tell you, because I told you when I first saw this enzyme, all that I didn't even know where the active sites were. So what if you look very carefully, which is not shown in any of the slides, and over the years I cut those slides, which is bad actually. You see this, I've colored it in cyan, and then I've colored it in a little bit darker blue. There's a gene duplication event which has happened over here. Okay, so what we realize is per ammonia only one active site is there if you calculate. Okay, and what has happened in the other active site is there is some some kind of regulation, which I still don't know properly, so I'm not going to comment on it. There's an ADP which was found bound without me ever adding ADP to it, with 3 magnesiums here. And when we first thought this was the active site. So there is with the simulation that we have done, we saw some motions. We feel that it can give feedback. Here, ATP gets converted to ADP, and ADP could go here. And there is a way that this helix could open and ADP can move, and some kind of regulation could be there. But we have still not been able to figure it out. But the enzyme is so big because there is a gene duplication event which has happened, where the mass of this protein has doubled, and only one of the binding sites is functional, and the other one may be doing some regulatory.
Thank you.
Okay. So,
yeah, sorry, Ruchi, just to follow up on that thing. In fact, that was the same point I was wondering. So if it's ATP, do you think ATP to ADP or ATP hydrolysis drives any conformational changes in this protein? Because that's true for other big systems.
So we have solved the structure with just ATP bound to it, and this thioester was still there, and we didn't see any major conformational changes. But very recently, in my last slide, I will tell you that we have tried to label this protein, and we do see a lot of changes. But that's our first experiment that we have seen in solution. So I will talk about it at my last slide, because now we are trying to do dynamics with this protein. So in structure, we did not see any changes. So we assumed there were no changes. But when we started doing, in solution with fluorescence tags attached to it, we see lots of changes that are happening. Okay, so there could be lots of stuff happening, and at that time maybe crystallography was not the right way to assume that ATP doesn't cause any changes. Structurally, we did not see any.
Quick question, Ruchi, before we move ahead. You're saying ammonia is released. Ammonia is the nucleophile that is reacting, or is it getting released from the reaction from this?
So first, in the first reaction, which is here, ammonia is getting released. Sorry, ammonia release doesn't mean it's going here. This is the active site. Okay. And here, then it trickles down in a blue. There was no path found when I solved the structure. And then it reaches this site, and here ammonia is consumed and added into the purine network, the growing, chain. Okay. So now what we found was that when that in certain organisms, 3 proteins, like I was saying, come together, and only when they add ADP, which was the question, that when the complex is formed. Otherwise, they don't come together. So ADP was not here in this protein. When I sliced it, ADP was bound to it without me adding it. Here, we had to add ADP, and the complex came together. So definitely, ADP has a huge role in forming the complex. But what ADP does more, I still don't have. Maybe in the future, in our 20 years of work, or 50, 20, we will know one by one. So, but this actually, although I say strategies for drug design, because there are some significant differences that arise in these complexes, and I'll tell you a little bit about it, maybe, or if somebody asks me, I'll entail it. But that could, we could, because this is present in gram-positive bacteria, and all humans have this protein. There's a huge difference. If you don't let this complex come together, they will die. And our complex is already together in place. Okay, so there are differences between the 2 organisms that ways. And I wanted to tell you that without having this QS, which is the adapter domain, no activity. Q is inactive in the until unless it is all put together. So it cannot produce ammonia on its own. No ammonia, nothing till all of the 3 come together. So really, their activities are interdependent on each other. So when Nandini took up this project, Nandini was thinking, she has done all the work, and now she's giving me this project. I told Nandini, nobody knows where the ammonia channel is. You are the person who's going to figure it out, and I'm not giving up on this project. So we started working on it, and we solved, like the question was asked, first structure was with ATP bound. We wanted to know what ATP is doing. Our structure showed nothing changed, and that was kind of a sadness for us, but we moved on. And what we found in the structure was this is the 1295 amino acids. The N terminus, the C terminus, these long loops, everything is ordered. Except this region right above the active site, which is called, we are calling it the C loop. catalytic loop. That is very odd that in a protein that everything else is ordered, and we were able to solve the first structure to about 1.7 angstroms. And this loop, which is 22 amino acids, was not fully formed. So Nandini started chopping this loop. Not only that, she took 500 purelles, large purelles, and she saw there was a high degree of conservation in this loop, which already tells you that there is importance associated with this loop. It had regions which were a lot of glycine and serines were there, and some aspartic acids. So then what Nandini did was, Nandini decided that she will divide the loop into 3 parts. One is a non-conserved region. One would be the region which is closing on the active site, which she thinks was closing using, we did modeling, and the other one which was facing away from the active site. So that's how she labeled it. We could have been wrong at that time because we didn't have a structure. So we wanted to know whether we can answer more questions than what have been answered in this, like how is these 2 active sites connected, how is the allosteric roles, all that Ankuna asked me, we wanted to ask ourselves, okay? And where is the tunnel, all this while that we are looking. So before Nandini also, we had dwelled into it, and I will tell you because in the story how we had collaborated with a group in Japan to answer some of those questions. But with Nandini, we made a lot of headway. So here, when Nandini did this, what happened? You take this blue region and you delete it. You have no problem with ammonia production. You have no problem with FCAM synthesis activity. This is the product. I mean, it's the reactant, sorry. Double bond O will become double bond NH. So there's no problem. So this blue region is redundant, to be very frank. Okay. But then you look at this red and this golden stars. When you mutate them, ammonia activity pretty much remains the same. You can see a little bit gone down, but FCAM synthetase activity is gone. That means this site is ready to produce ammonia. It is talking and everything. Otherwise, the site is not producing ammonia only, right? But now it is producing. But now this site is unable to turn over the product the way we want. Okay. But look, the most interesting mutations are these black mutations. Something has happened, some stability issues have gone, and this substrate is not properly getting anchored, it looks like. But you look at these black mutations. Black mutations, as you are making progressively, you can see that ammonia activity is gone. These are 30 angstroms away from the ammonia binding site. Obviously, if you make something on top of this site and it goes away, you are not. You say, okay, it's right adjacent to the active site. This one is 30 angstroms away from the binding site, and yet it has had such a profound effect. Okay, so we also did ITC on these mutants, and we got this. What we surprisingly saw was that the way the activity is getting shut is because as you make these progressive mutations, this site cannot even bind glutamine. Something has happened that this glutamine can not enter the site, and it is completely shut because it is not able to get glutamine access. So to an intermediate state where the glutamine access was there but not that good, and the activity had gone down, we decided to crystallize it. And we were lucky. We were able to get a catalytic loop completely closed in, and we could get glutamine, not thioester, and we were able to see this binding site. Okay, so now this loop has been closed and it has suddenly formed a structure. It has formed a secondary structure, and a previous structure we had solved also had in a standalone place this loop closed with F-gar bound. Okay, so now we could see that the loop has closed on top of the active site, and we decided that with this, and we got it at pretty high resolution also. We decided that with this we will do simulations with Jagannath. Jagannath did 1 microsecond simulations, and I'm going to show you what we saw in the simulations. We saw during the simulation, this loop starts opening up slowly. Okay, and so from a very close state, it starts opening up. And as it opens up, what happens, the relay that happens during is what we were looking at. And instead of this, which shows the conformational changes in more complicated way, I'll just show you a PCA where it has been just enhanced for you all. And here you can see the regions, which is the N-terminal domain and our loop and this linker region, they move a lot during this process. as compared to the 2 active sites. And so what we did was we looked at how the C loop is opening. How is this opening up? Okay. So this interface, we made a movie some more, I would say, from the simulation, that as this C loop opens, what we saw was that this domain kind of closes down. I think ultimately it may close up, but this end terminal domain, it pushes, but it moves around 8 degrees and pushes this domain. Okay, that's what is happening. This loop is opening up. So during catalysis, the loop is closed, and during the progress of the catalysis, loop opens, and then it makes a dynamic interface with this end terminal, and then it goes to this one. Then what? How do we prove it? No, it's a simulation. I'm not supposed to believe everything on a simulation. So we said, okay. Remember, we had mutated those 3 black mutations. We wanted to know the black region, how it interacts with the end terminal domain during the simulation.
Ruchi, can I ask a quick question?
Yes,
Sorry.
yes.
Can you go 1 or 2 slides back where you had the RMST? Like, yes, these structures. No,
yes.
The next one. The structures together. नहीं जो एक साथ loop के dynamics दिखा रहे थे ना? You had stacked all the structures together. हाँ, इसके बाद, yeah, this one. So in this thing, we can see there is a lot of movement in the yellow part, right?
Mm-hmm.
And there is this chocolatey brownish part as well, and the red.
And the red.
So that is where that loop is basically.
That is the C loop. The red, unfortunately, the color got changed.
Okay.
And I had to make all the slides with a different color, which I did not, but I did not do that.
That's the C loop.
That's the C loop.
Okay.
That is the C loop.
And that is the one that opens, that is closed in catalytic cycle.
And then it opens up.
Then it opens up.
And as it opens up, it hits this end terminal interface. And then this end terminal interface starts moving and going to the glutamine. Moves up.
Where is the energy coming from? All of this is thermal energy? Just thermal energy?
No, I mean, see, when it cannot be just thermal, it is during the catalysis, it is the hydrogen bonding networks are changing.
Okay. So it's part of the fluctuation of the thing, but it's during the catalysis gets.
It is like a new, when the reaction happens, it's an ATP dependent reaction. And when the reaction is happening, there is a push, I guess, from the reaction because the substrate is changing in some form. And that push changes the hydrogen bonding network between these.
Okay.
And some hydrophobic networks, and that results in various changes that happens.
Okay, thank you.
Now, I mean, if it is only thermal energy, then this enzyme can work all the time.
Okay.
Right.
Okay. Yeah.
But we are able to see that it is trapped. If you give glutamine, like in 10 millimolar access, one molecule of glutamine just goes and traps itself here and never comes out.
Got it. The substrate has to be there. The substrate has to be there. The catalysis has to be on. Then only it will open up like this.
Yes,
okay.
Yes.
Fine.
And also this glutamine site, if you in proteins, which you leave it alone, it is unable to do anything. It's just like a very bad enzyme. After a long time, one ammonia molecule comes.
Right.
And it's just like, not working.
Okay.
Kind of. Okay. So what we did was to confirm, let's say, the results of the simulation. What we did was we said, let's look at the changes that happen in C-terminal during the simulation, and we saw that new connections are being formed as the C loop is opening. Okay, and these connections, are they important or not? We wanted to know, or just fiction of our imagination. So what we did was we looked at those mutations, like I said, the black. And we realized that correspondingly to that area, new connections were being formed. And we left the C loop the way it is without changing it. And we made mutations in this red region where the C loop is contacting the end terminal. Okay. And I have only shown the result, which is the final result here, but we made 1 by 1 mutation. And we saw identical result to what we see for this one. By the time 3 mutations are made, no glutamine binding is there, and complete loss of activity at the G site, which is 30 angstrom. So this time our C loop is intact. None of the black mutations have been made, but the black regions or close to the black regions which contact the N-terminal region, which we saw in the simulation, that we have removed. Okay, so now we have more confidence that what we are looking at is actually real. Really, the signal is going from here to here. Okay, and really, if you leave the C-terminal loop intact and you get rid of the dynamic interface, you lose activity. Okay, at this site. Okay, and it is like 100% loss by the end of the mutation. It's not like little bit is left. Okay, so and there is no glutamine binding again. So then what we did was we kind of realized how the signal most likely is passing through. But then we wanted to look at this interface. What is happening at this site? Remember, this glutaminase domain is not active by itself. So we wanted to get some clues what is activating this glutaminase domain. And so we wanted to see the passage of the signal. So here, even now, we have not done all the mutations because we are so caught up in so many things that we are doing. But I can tell you, during the simulation, there is what we see is there is this release of this place called oxyanion loop. So many of the glutaminases have this oxyanion. I'll talk about it a little later. Just give me 2-3 slides after I introduce the channel to you, which was supposed to be happening. And that I will talk again because we have done more work now on it. But basically, you can see that this region in green shown in here is getting, and here you can see it was kind of connected and it got disconnected when this N-terminal domain started pushing it. And so now this releases this oxyanion loop, this region, which is very important for activity of the glutaminases. And also, if you look at the passage between this region, we see that the passage opens and closes. So glutamine cannot enter because if you close the passage, then glutamine cannot enter. And while this is happening, the allosteric signal is passing, the passage kind of opens up and most likely allows glutamine. But if some mutations happen in the N-terminal, the passage will not open up and access to glutamine is kind of gone. Because you see, every time we see that glutamine binding is completely gone. So we wanted to see if that is the case. So we can explore that aspect also a little bit more. But let me come back to what I talked about tunnels, right? So we know the allosteric signal is passing. I have talked about it, but one thing we don't know is how is ammonia traveling from site 1 to site 2. So when we first saw the structure, we looked through spaces like Jyotishman had asked me, and I tried to find on my own. And this is my version of it in 2004. I thought there were 2 channels. I had no clue where the channel was. And I just looked at spaces and there were blocks. No channel was complete in the structure, but just to make guesses little bit. I mean, I went mad looking at the structure, I would say. And I came up with a direct route and I came up with this route. Okay. Now, since I told you I was pretty stubborn on trying to find out the channel, we collaborated with Dr. Hirata he was in Institute of Molecular Sciences and he moved after retirement to Ruchi. And I told him, nobody's ready to do the simulation. It's a 140 kilodalton enzyme. What should I do? Everybody says it's too big a system. The computers haven't developed enough, or nobody wants to give me that much time. And within 2000, we started much earlier than 2015. And it's an interesting story. We met in Mauritius, and I kind of heard his talk, and I said, maybe you can help me. And then he was like, okay, let's try to work. And I went to Japan, and he got convinced. I mean, he called me. And what we found was that out of the 2 channels that I showed you, one of the channels using Wilson theory, which is an analytical theory, uses StatMech mostly, interaction site model. And here, by looking at interactions of the protein with ammonia, they found that channel 2, one of our channels was, if I go back, which is this channel, which is near ADP binding site, is the highly likely channel, most likely channel. And they also did mutations along the channel, and they showed that generally the way the channel is, it's 23% success rate of opening of the channel. But some mutations can really make it very bad, and other mutations can, for example, make it 88%. Okay, so we I will come back to this work. This was very helpful to us actually, that how to reduce the channel 4 and increase the channel 4 and to decide whether it's the channel. But more recently we worked with Jagannath, and here when we did this simulation, we saw the entire channel opened up. Okay.
How does this compare to, say, the known ammonia channel, which was there are known intracellular, like on the membrane channels with ammonia going inside the cell?
They are very different from membrane.
They are very different
from ammonia. In there, the ammonia directed like through some kind of a relay, which is hydrogen bonding relay.
Right.
Our channels are very hydrophobic.
Very hydrophobic. Okay.
Yes.
So can they? So I know ammonia size. Will that match something like a metal which can be put in, because there could be enhanced scattering factor and you can look at.
Metal will not go in. That's a very good idea, but not going to be applicable. But we did something else. We tried to match it with xenon.
Xenon, yeah. So something like
xenon is slightly bigger, but krypton I was unable to do because somehow. So I have a paper in class 1 where we tried to do xenon, and we were able to push xenon in the top of the channel, and the rest of the sites it went was all here and there. Now I know. Okay. Went to other places. Okay. But one of them went to the top of the channel right there. Now I'll tell you. But the krypton is the right size for ammonia. But we tried xenon and then we could not try krypton. We tried to flush it under high pressure of xenon, and I think krypton would have worked. And krypton also has something called a mad signal. So you can actually do multi-anomalous scattering, and they'll light up.
Right. Okay.
My dream remained a dream. So far, maybe it will again. Now I will. I mean, you need a setup for it. And one of my collaborators, Santosh, had a setup.
The other part is some NMR folks actually work with hyperpolarized xenon, and they look at signals.
So xenon is not good. That's what I realized. But krypton would be
good. Okay.
Because size of krypton and ammonia is very similar. Okay. So then, anyway, we still need to do experiments in that area. So we are lagging behind with that area. So here, what we did was the whole channel, it's separated right where I had predicted, which I feel so glad about, and where Hirata had predicted. That's exactly where the channel turned out to be. Okay, and I will show you more evidence in our favor, because we at that time did a crystal structure in the middle of the channel and blocked it partially. But I can show you that if you see the ammonia, there are gates that I'm going to talk about now. There are two gates, a mouth gate and an end gate. This histidine you can see is opening up, and this arginines, and this you can see this flipping of things are happening over here. This serine is going away. So there are gates which are there, which are at the mouth and at the end of the channel. And then you can see, I will show you because now I'm so used to it. This moves a little up. This distance goes here. This B moves in some way. It's a simulation. You don't have to believe everything, but if you believe me, partly also. The whole of this beta sheet moves back actually, and this helix turns this one and opens the channel. Okay. So we were able to see this channel. That was something that was interesting. And to my support, I would like to tell you that remember I showed you in the middle of the channel this V is there, and we also got this rate of V 13%. The channel is closed. We were able to make the mutation and solve the crystal structure and show the constricted channel. Actually. And in that case, we saw that a lot of leakage of ammonia has started to happen, and the enzyme was about 20% of emission. Okay, so it is in the middle of the channel, but ultimately the efficiency at the FCAM synthetase site went down, and there's a lot of leakage of ammonia that was happening. And the other mutation that we wanted to really construct the channel, every time we tried this mutation, we ended up making the protein completely insoluble. Okay, but any mutation which made the channel bigger, we realized only resulted in that particular thing to have a rotamer which goes out. Activity was not affected, and that is because the rate of production and consumption is not getting changed. So middle May, if you don't stop it, it cannot enhance its activity. So nothing happened to it. It was as active as our native. So
Also, Ruchi, sorry for disturb. Ammonia, phosphine, arsine, all are in the same series in a sense. So, but the sizes go up. So, is there a size restriction? Is there a?
Yes, the channel is like really not very big.
Okay.
Actually, I will tell you something else, which I will show you in like 2-3 slides as we end, which has some implications on why ammonia. I'm not sure, but that's my theory. Okay. So let me continue. What we did was, if you remember in my previous slide, that with the movie on, these gates were there. So we decided to concentrate on these gates now and see what is the regulation that is going on. We are able to see the ammonia path, but are these gates, are they controlling? How are they controlling the whole process? Okay. So if you look here, we have arginine. The top gate is a network, like an aperture type of gate. Okay. And the other one is just like swinging gate, door open type of gate. That's the bottom gate, which is our histidine gate. Okay. So if you look at the top gate, now you see arginine, serine, aspartic acid. This is from the F-GAM synthetase domain. Remember I have this coloring, this complicated coloring. This is from the ammonia amido transferase domain. And this N, aspargine is there. Okay. So these are the gating residues which are there. And what I want to tell you is this arginine, when you mutate, we did alanine scanning mutations. And when you mutate, suddenly we see the enzyme starts producing more ammonia by 25%. And in case, if you make arginine and serine mutation, it also becomes more hyperactive. The enzyme becomes more active than the native. Okay. So these two mutations are helping the enzyme, it's becoming more active than it was in its original form. But if you make mutations in this D and this N, in one case, it becomes hyperactive, ammonia production, but becomes really bad when it comes to F-GAM synthetase. So it's, and if unfortunately I'm not able to see my last slide, but you can see leakage. I moved my view. Leakage has gone up with respect to the native for some of them. Okay, so this is less leakage. That means ammonia is being produced and completely getting utilized, and the whole activity is going up. But here, ammonia, even if it is produced like in this mutation, it is not getting used. Okay, so something has happened in this, in these when we are doing this. When you look at this end gate, I will describe some of these mutations later, but the idea was to perturb the end gate or close the end gate permanently by making mutations between hydrogen bonds with the end gate. Okay, but if you look at anything we do with this end gate, if we try to close it in like a tight way, no ammonia production, no F-GAM synthesis. I told you before, this is 30 angstroms away, and I'm making mutations down there, and the end gate seems to be like a super master controller. Here, the activity may go up, go down, but here, dead. Enzyme is dead. This histidine, you don't even mutate the histidine. You keep the histidine the way it is, and you say here you have mutated the histidine. Here we have not mutated the histidine. All we did was we looked at strengthening the hydrogen bonding of this particular gate, and the enzyme just goes dead. Okay, so this is the difference between our two gates, the controls, and the level of controls that it is giving. Now we wanted to know this arginine, why removing the arginine is resulting in enhanced activity. So what if you look at the binding site, what we did was we did two sets of mutations, like I said. We made this S to a D, so we reduced the charge on it. In one case, we increased the charge by removing this D. So when you what we're trying to do here is we're trying to concentrate in this region, and we are going to make mutations of arginine, serine, N, and like I told you, the way I told you, and this D, one by one. And in our bigger picture, we also want to know something about our oxyanion whole ordering, if we can tell more about it. Okay. As, why I just told you in this channel, these are the gating residues. And remember previously I told you this loop, oxyanion loop, and connecting at the end of the oxyanion loop. This is G. These are the residues in the oxyanion loop. Okay, hole, which form the hole. And it is this flipping of these G's they think is important for stabilizing this anionic state or not stabilizing it enough. Okay, so when this flip of these two G's happen, then only you will see that the transition state, where the backbone of the peptide backbone is getting stabilized, this anionic state. So this is the general story, but let me go back to the first part where arginine is increasing the activity. So we tried to see if it's a steric effect or a charge effect. So we kept arginine there and we mutated the serine to a D. The activity still was 25% more. That means arginine did not lose its place. If it was a steric effect, you would not see this happen. It was not, so what we see is, but by putting it as a D, we changed the overall charge on the arginine. So you can see here ammonia is produced and it has to go in, and it is hydrogen bonding with our arginine. Okay, so what we think is we solve a series of crystal structures with these single mutants, and this is the native and this is our arginine mutant. And you can see here in all, and this is the one which is the S1052D. So here, obviously, there is nothing, no arginine left, and ammonia can just go because this charge effect of R is no longer there. But if you look in this one where the arginine is still there, it is now hydrogen bonding with this D, and so the charge effect has gone down on it. The arginine is no longer that much positively charged, and so it is not controlling ammonia. It could also be participating, for all you know, during the transition state. It's very close there. It's not a catalytic residue, and it may be influencing ammonia release itself. And we have to do some more calculations for that, which we haven't done yet. But we predominantly feel that it is a charge effect, and it is the arginine, which is like a controller residue, which is helping in ammonia going down. Not only that, when we have seen that when we get rid of the D here, which is in this, the arginine has its full force on this ammonia, and you can see that both ammonia production as well as ammonia, then obviously the other reaction goes down. But the most interesting mutation we saw was this N1051 that I talked about, which is it is adjacent to the oxyanion hole. It actually hydrogen bonds with this oxyanion hole residues. And what we saw was, because we had very high resolution structures, that in part of the structure, we could see the oxyanion hole flip in. And that is what is supposed to happen when you are actually, first there's a release which happens, which is because of the allosteric cues, and the oxyanion hole actually is able to make it conducive environment for it to now serve as one. And when you make this N1051 mutation, this hydrogen bonding is lost between them, and this becomes flexible. And you can see when this happens, it has enhanced ammonia production. So this plus is telling you that ammonia production is more than the native in this and even in this. But it can produce ammonia at a higher rate, but it is unable to consume it at a higher rate. So what happened? So if you look at these crystal structures and look at how they're connecting to the tunnel, what is happening is that this tunnel, when this happens, if I show you, although I keep showing this, you can see that as this D is moving along with this arginine, this hydrogen bond is breaking, and it is moving away from this A384. This bond is breaking, and here also you can see this N1051, which is this which I'm pointing out, is connecting to the tunnel, and it is also you can see some small motion that is happening over here, which is the motion. You see this one goes through. If you look here, you can see that this is the oxyanion hole. Okay, so what is happening actually is that these there is a way in which the tunnel opens, and the D65, the arginine 1263 rotation allows D657 to talk to no longer talk to A384, and the signal goes down. And same N1051 talks to this N378, and the signal goes down. And when you disrupt any one of these interactions, which will be between closed and open state, then the tunnel is not able to open or close effectively in both these cases. And so the signal is not passed down properly for the tunnel to open effectively. And as a result, the whole process becomes slower. Now, when I talk about this, I would like to say that we wanted to investigate and see if this kind of gating network is there in other ammonia enzymes. And to our surprise, although people have not pointed out so much into it, we found that versions of this gating network is present in class 1 and class 2 amidoclasterases, where there's an arginine or a lysine residue, or you can see in many cases an arginine, which is there, and there's a serine, like I said, everywhere. There is a D, which is going to the tunnel everywhere. There's a N1051, which is there, which is in our case connecting to the tunnel, and it is present as a Q or an N, which is same amine moiety is there, amide moiety. And you can see in most systems, in some it has moved into the actual oxyanion loop itself. So in these cases, you can see it as this N residue has moved inside the oxyanion hole, and it forms local hydrogen bonding and changes its environment, and so that it is activated. But it looks like that this mouth gate type of network is a ubiquitous phenomenon, and not just us, but other systems also control their ammonia production through this mechanism. Now, why this ammonia production needs to be controlled is a controversial question that came to my mind also. From my side, it looks that I'm not 100% sure we now are doing coupled kinetics, but it looks like that the F-guard synthesis reaction is slower than the glutaminase, which is a very highly evolved reaction. So maybe you don't want to push ammonia too fast. So it may be regulating this fast enzyme because it is now actually bridging this enzyme with what is going on. Okay, so that is a conjecture. I don't know, but that's what I thought. Okay, so coming to the end gate. When I told you, remember I told you that we wanted to close the end gate permanently. And here when we made G31S, which is now capable of hydrogen bonding, and T3TN, which is now better hydrogen bonding distance, I mean, this is like you can get it to be tighter. We saw that again, glutamine binding is gone. And this is completely in 500 plurals. This is completely, the end gate is very conserved. The mouth gate is also conserved, but the end gate, it's like conserved to death. I have to say, like not even one fluctuation happens. Q to N also doesn't happen, kind of thing. Okay. So what we realized is that this gate opening is very important, because if the gate doesn't open, 100% loss of activity is happening. So we wanted to know how the gate is opening. So what let me tell you what we have snapshots of. Here, H296 this is the gate. And then when this FGAR comes in, which is a substrate, this actually acts like a proton donor in the reaction, we feel. And that is how the gate gets opened up. It moves towards the FGAR. Okay. And then this aminophosphate intermediate will be formed. And there is another residue which comes close at this point, which is I'm showing here this glutamine, which hydrogen bonds with in this state when the gate is actually open with 2471. Okay, which we saw in the simulation. So and then we saw that during the simulation and in some crystal structures, this moves a lot. It moves in and out. It's a C loop. This is a C loop. It is on the C loop, this residue. So it talks to the H296. The minute this FGAR comes in, there is a contact between them, and it as the reaction is going, it tells the progress of the reaction, and the C loop starts opening. So if we mutate this, we see about 60% loss in activity, because this is a pretty big loop. So one residue changing, it's apparently 60% loss of activity we get, and that's after kinetics, not single point. So that's how we think the C loop is getting a signal how to open. When does it know how to open and what causes it to open? And this is just showing that it is, you can forget about it. This distance is increasing, and this distance, it's moving towards the end terminal. So we looked at its progress towards the end terminal, and during the trajectory, it goes very close to the end terminals. Okay, and because a new interface has to be formed at this point. So this is our end gate, and this is almost my final slide. What we noticed during our simulation was that, remember I showed you the tunnel. When we peered into it, we realized that there was a closed state of the tunnel and there were a couple of open states which was fluctuating. And when we looked very carefully, what we saw was that this phenomena was this. There are some missing residues. I could not make a new figure, but you can see that in the tunnel here there are residues which are partially closing and partially opening. So there are portions of the tunnel where it is not completely open, it is not completely closed, it is partially open. And it's partially, so what these are showing are distances where it is showing when it is closed, then it opens, then it is closed, then it is open. So it is like a vacillating tunnel. It goes open, there is a closed period where it is closed, then there's an open period, and then it goes like this. Okay, and so slightly closed, slightly open, slightly closed, slightly open. So we feel that this motion could be the reason that you can, if it transmits down, it can be the reason that it is kind of pushing ammonia down and going into the tunnel as it runs, and it is not like it's open all the time. It opens and it closes. And so it's a fast motion during the cycle. So in the closed state, it's completely closed. In the open state, it is like this. Okay, so a little bit closer. These distances have been shown here. Little bit far away, completely open. Little bit closed, completely open. Little bit closed. Okay, and this is just showing this trend in these residues. N30, some of the residues which are on opposite side, and some of the residues which are the D657, which I showed you. This one, D657, arginine, this A384, this part I'm showing you, and some distances. Okay, that we feel in the open state it is happening. So it's like a pulsating tunnel, and that could be one of the reasons we feel that many tunnels may be pulsating. For example, mostly gaseous oxygen transport or carbon monoxide transport, they have to go and they cannot interact with the through hydrogen bonds that easily, because they have to be maintaining their chemical nature. So they many times, maybe just to make sure it doesn't go anywhere else, doesn't go up or down, it can just squeeze it, it closes it so that it only goes in the back part, and no reflux happens, which I've been facing a lot. So to kind of prevent reflux from happening. And it is known that more than one ammonia is produced in a cycle. About 3 ammonia are produced per reaction. So I told you that reaction is much faster than, but 2 are wasted, goes out. Okay. So lots of, when you see the enzyme in its basal state, you would think, oh my God, it is active, but it is much more active than it can get. So that's what I wanted to pass the message here. To continue with our ideas, now Sukhvinder has taken up, and we are now incorporating unnatural amino acid into the protein at various positions. We may even try
Ruchi,
to. Yes.
Can I ask one question?
Yes.
Yeah. So Ruchi, I'm just trying to correlate your tunnel with like a pores in the material, which controls the flow of reactant. Here, they are controlling the flow of molecule towards active site.
Yes.
So is it not possible to use, the neutron scattering and xenon NMR, combination of them, which generally gives excellent information about the pore size, the pore shape, and how they are connected. So it will be the same.
Neutron scattering, we need much bigger crystals. We actually tried neutron scattering, and we actually even sent it to ASRF with our crystals. We need crystals for our particular, for our system. Because it's a very big biological system with 1295 amino acids. Same problem is with NMR. Okay, we want someone to work on it. I would love it, but they are so scared of the system. We were people who were scared of simulating because it's so big. So the same problem is with NMR.
But recently,
not xenon, but a little bit smaller than xenon. We can find something.
Yeah, because
xenon would be good.
Xenon will give us a more accurate.
If it goes in, if xenon is slightly bigger than the pore. That's why I was telling you about krypton.
Krypton, then the neutron is the best. Then.
It will be very good if we could do that. We could also start the reaction in some way and look at it while the reaction is happening. We have so many mutations happening that we have slowed down the reaction in some cases.
So we Ruchi recently did it in bark. Bark also has this system where they must be saying.
We are unable to do for proteins. That's what I'm saying. That's why I went to ESRF. And in our case, they could not do it without crystals.
Okay, but ours is a amorphous material, right? It's huge, 500 nanometer, far bigger than yours, but they managed to. Then I may be
because
talking
the repeat unit is not so big. You understand? Our repeat unit is 140 kilodalton. Repeat unit is the problem, not the overall structure. Possibly, our repeat unit is the problem. Repeat unit is this entire thing, and not just. It's you understanding what I'm saying?
Yes.
That's your conglomerate is made up of some small repeating unit, which is there. But our conglomerate is made up of unique things. That's why it has become so difficult. The interactions are so many and so many possibilities exist that people are not able to get data which they can make sense of.
Yeah, can provide.
That is where the problem is.
Okay.
But yes, any kind of these experiments, NMR would be very good, I think, if we can do with someone. Because we have the structure of the protein now. So in the app, what we decided was like similar to, we have the same ideas, and any ideas are always welcome. That we will try to do thread-based work. Okay, when we started thread-based work, I thought about it, which may not work actually in the end. But what we thought was that how to uniquely even have our protein labeled, because there are so many cysteines that you can't find. There are so many tryptophans you can't find. How do you label arginines which are encountered? So then what we did was we decided to use something called unnatural amino acid machinery, where we can put it wherever we want. We need to get 2 unnatural amino acids. We've got just one working now, and the other one, when lockdown happened, we were in talks with this guy who is in Peter Schultz's lab. And we have got this also from one of the Peter Schultz's lab. It's a gift. Okay. And in this one, this is one of the amino acids we can incorporate. There is a collection of amino acids. But we used coumarin because we wanted to do some initial studies to see whether our machinery is working, and you can see it. So coumarin absorbs, emits in the visible, 450 nanometer range. Okay, and you can see our protein, it's incorporated. You can get it sequenced. So then it is only one fluorophore in one position where we want it, not anywhere else. And now what we have done is we have put it in the C loop. Remember that I told you C loop opens and closes. So currently we have only put it in as this is a future direction. And we have only put it as a test in one place, which is in the C loop. And already, like I think Ravi asked me that question. Already we see that this is just very 2 days ago. Actually, this is really something which I was not even including in the talk. Then I included, where we see that there are specific states where you have just the protein plus ATP, completely different, dynamics, and protein with FGAR, which are the different dynamics, and the state which is just the free state. And so we see huge changes in population and things going. And this is very preliminary data we just got from Sukhvinder just got it. And so now we also need to make a pair, which will be, and we're going to move this pair throughout the protein and see if we can capture dynamics with our knowledge that we already have on the system. So that is what we want to go. We want to track these states where we start with a state where the enzyme is completely not doing anything. Then the substrates come in, the loop closes, and then the signal starts getting transmitted as the reaction is proceeding, and the tunnel is formed, and oxyanion hole is actually rearranged. We want to see all these happening by some method or the other. And yes, the tunnel will be the most difficult only by thread, but you could also do outside tunnel residues and see if you can see something that I'm saying. This is one method, but yes, NMR is the other gold standard method which will work if we are able to do it efficiently. NMR, people with NMR, they can really help us in tracking this down. And neutron scattering, I told you the problem we have, but there we can look at protons in our system.
Yeah. Because neutron can also tell you the leaching path, because you are saying some of the molecules are leaching. That means I see that as a very small pore. Maybe
so when we leach them, we actually what we do is they convert into ammonium, and then we just monitor how much ammonium is there, and we have how much glutamine, how much glutamate production, how much ammonium we measure, and how much FGAR production, and from there we say, and generally the enzyme which is purell by itself leaks. we just set it to 0.
Okay.
And then with respect to it, we should say more leaky or less leaky. So when I show those bars, it is with respect to the Purell, but Purell by itself leaks.
Okay. Thank you.
Okay. With this, actually, I'm going to end my talk. And Nandini was the major player, I have to say, in most of this work. And Sukhvinder has taken over the project now. Padmini helped a little bit in the simulation. And without Jagannath, we could not have done these simulations. And Navjeet was a big help with Nandini. Actually, Nandini was like the forerunner, I have to say, in the thing. And she even learned all the stuff from Navjeet to be able to study the simulations in such a good way. And Navjeet had a lot of knowledge, I have to say, and he helped us a lot. And Jagannath and Navjeet were very instrumental in our project. And we have a ongoing collaboration with Jagannath in almost all our projects when we come with the simulation. The project I was talking about, the ATPAs, is also something that we are exploring with Jagannath quite a bit now. So with this, thank you all for listening to me patiently. And I hope I was able to convey this topic in a much more lucid way. Thank you. Thank you so much.
Thank you. Thank you. Thank you, Ruchi.
Thank you.
Would like all the audience members to unmute themselves and just give a round of applause. It's something that is usually missing in these Zoom seminars. Unfortunately, you don't get to hear the claps. So thank you, Ruchi. The talk is open for questions, more questions from the audience members. So please unmute yourself and ask. Yes, Mitradeep. Mitradeep has raised his hand. Mitradeep is a student, second year PhD student. Mitradeep?
Hello.
Yes.
Yeah, so thank you. It was a really nice talk. So I was wondering about the allosteric interaction you were discussing with the C-terminal loop to the ammonia binding region, which was around 40 angstrom away.
Yeah.
So can you please talk a little bit about how the information is transmitted?
Okay, in this one, I will try to tell you, although it's a schematic. What happens is that the C loop is kind of disordered, and then in one of the sites, our substrate FGAR comes in along with ATP. At that point, the minute FGAR comes in, this loop has interactions with FGAR. Remember, some mutations we made. It has backbone interactions with glycines, which are there. There are glycines in this, which actually interact with the substrate. So this kind of orders the loop into position. But at the same time, the minute FGAR comes in, the reaction is set into play along with ATP. This histidine, which is in this conformation hydrogen bonded with this threonine, it swings towards FGAR. It has, when the FGAR is not there, it is completely, with the threonine. But the minute you make it swing towards the FGAR comes in, it swings and helps mostly think in proton donation during the reaction. And then this gate opens. This is our end gate. At the same time, this loop, as the reaction is, the first immunophosphate intermediate will be formed. ATP will become, we will make a phosphate onto the substrate. And when that happens, there is the loop starts opening up and it goes and hits the N-terminal domain. And the N-terminal domain, which is labeled in red, unfortunately here, it then gives the signal to the glutaminase domain to restructure its active site such that it can start producing ammonia. At the same time, the tunnel also forms. And the whole ammonia release happens because now this restructuring of this loop happens, so ammonia can be produced and it reaches here and the reaction cycle is complete. Is it a little bit better?
Yes.
So basically, the trigger of the whole cycle is with the substrate called FGAR, which is the.
Okay.
I show you the structure. Getting better. This is the.
Mitradeep, move away from the mic.
Oh, sorry.
This is the substrate.
It's like Wolfman.
Then when this enters the active site, this particular compound, when it enters the active site, it pulls the bottom gate with it. Okay. So when the substrate I showed you enters active site, first it orders this loop. You can see that this loop gets ordered.
Okay.
And it also at the same time pulls the our gate which forms the tunnel, which is here. This is pulled towards the F guard.
Okay.
And this opens the tunnel in the bottom, and this F guard also, closes, coils up the loop, and during the reaction, it opens up and gets the signal through the N terminal domain, and then ammonia is produced. So it is a stage-wise process. First,
Okay.
F guard will come, then it will pass the signal, it will open the gates, and then it will go and ammonia will be produced.
Okay. Thank you.
Any other question?
Yeah, I had one actually this time.
Yes, Ravi. Yes.
Yeah, Ruchi, so great talk. Always enjoy your talks. So I'm glad this tunnel work has reached, such maturity at this point.
Yes, yes, Ravi, I know.
Yeah, I'm intrigued by this second ADP, which is stuck at the other, side of the thing, basically. So that ADP came without you actually putting in any effort.
At least in the crystal structure.
Right. So if you actually really go in that direction, and let's say you incubate with ATP, right?
Yeah, we incubate with ATP all the time for a crystal structure. ATP was like in high excess, and still that ADP remained. When we saw the structure with ATP bound to the protein, only ATP bound, the ADP was still there. The ADP is stuck there. The way until the conformation of the protein changes, ADP doesn't come out. So you need maybe F guard also, ATP and F guard. Then maybe that ADP may just move.
But there is no space, you said, for anything else other than
in ADP. That ADP side.
Yeah.
Yes.
Yeah.
Yes.
So that's why I was thinking. Is it possible that there are other conformations of this protein and ADP?
Yes, that's what I was saying. What I was saying was we think, so in the glutaminases, there is this, can you see these two helices that I'm pointing out? They are extra domains which are not present in other glutaminases. They are just capping this ADP side. Right. And we think that when the glutaminase functions, this may actually move. And ADP may actually, there may be some regulation that is happening. And in our simulation, actually, we saw one of the loops, it actually moving from here towards this place. Some kind of motions were happening, which I did not show you. But if you look in our science advance, there is one figure which is on ADP also, which shows you that there is some communication happening between this site and this site.
Right.
But I didn't, we have not done any mutations yet or, exploded. So we left it with just a report there. We have to do some more work there. I mean, I think this enzyme is like a mine that we keep on working on.
No, very interesting facet. In fact, that ADP, the second ADP is also close to your channel.
Yes, it is actually forming it is close to the channel, and one of the things that is moving, it is very close to it.
So are there any regulatory elements which are close to that point where the ADP is closest to the channel actually?
I mean, we don't know. I told you what I think is this green region will completely open. It will move. Because this is not a normal thing which is present in other aminotransferases. It is just present in our enzyme. And I think it has a purpose. In the beginning, when I saw the structure, I always used to label it with a different color because it didn't match the aminotransferase thing. And that is right above the ADP site, and it may have something to do with some tunnel regulation also for all we know. We just don't know.
Right.
Yet. But if you mutate any of the ADP magnesium ions, the protein became insoluble. I remember that. Roughly.
Okay. Now, one final point here for the same ADP actually. If the ADP gets hydrolyzed to AMP,
no,
that's it.
ATP gets hydrolyzed to ADP, and that's it.
But after that, there is no possibility of the ADP getting further hydrolyzed because there's additional energy that can come in. And
as the next phosphate, maybe the active site is not aligned distance-wise to have a shorter. I mean, you have to rearrange the distances in the ATP has to fit, the other has to fit, and then the transfer happens. So everything that is aligned in the active site. So if you have an AMP, the whole distance matching goes bad in the active site.
Right. So I was, but the hydrolysis may drive, some conformation.
Yes, because we think after the immunophosphate is formed, this loop opens.
Yeah.
And ammonia is still not produced till it reaches this. The signal then immediately goes and ammonia produces. People have shown that the immunophosphate intermediate is a long-lived species in the absence of ammonia.
Mm.
So yes, it could be driving it to some extent, and that is maybe how this whole trigger happens, like Jyotishman was asking.
Yeah, very interesting system. Thanks.
Thank you, Ravi. Thank you for the question. Any further questions to Ruchi? Okay. Well, Ruchi, I think this was fantastic. Thank you for doing this colloquium with us today. And I'm sure, the audience here, based on the number of questions you received, was really excited about this work. So thank you very much. And I would like to also mention that if you were here physically, we would have taken you for tea after the colloquium. That's just part of the tradition.
Okay, I can't drink tea right now.
Right. Anyway, I know the situation you're in, but thank you. Even in this situation, you're doing this. So thank you.
No, I'm much better. I told you. So
Okay.
I would have told you.
Okay, sure. And I just wanted to thank everybody in the audience for joining in today, both through YouTube and Zoom. I also would like to mention that next week we are taking a break from the colloquium because of physics interviews. But after that, we are going to return with another structural biologist from ISER Pune, Sai Krishnan Karaya. Yeah, and he will be speaking to us on the 16th of June. So thank you again for joining. Thank you, Ruchi. And I will like to sort of stop